Hello, everyone, and welcome to phyloseminar.org. The current theme is pandemic analysis, and this is the fifth talk in a series of 5 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Nicola de Maio. Nicola did his PhD with Carolyn Kosiel at VetMed Uni Vienna. His main interests are phylogenetics, population genetics, molecular evolution, and genomic epidemiology. Nicola is currently a scientist at the EBI within Nick Goldman's group. Welcome, Nicola, and thank you for participating.
And thank you so much, Eric, for having me. So I will discuss about phylogenetics, in particular in relation to SARS-CoV-2, and more in general to genomic epidemiology. So as we have seen the last couple of years, phylogenetics and genomic data more generally can be very useful in this context. For example, we can use it to identify new variants and calculate how fast they're spreading compared to others. We can track the spread of the virus in a local setting, so in a school, for example, or in a country, or between countries, and so on. We can measure how effective the containment measures have been, and we can identify regions of the genome that are evolving faster or more slowly, and that's also useful for antibodies and vaccines development. And we can basically study how the very beginning of the pandemic, when there was not much sampling going on yet, by using phylogenetics, we can get information about the origin of the virus, for example. And the limitation here is that despite the availability of millions of genomes, the bioinformatic tools we have only allow us to study, most of them at least, allow us to study only a few thousand genomes at a time. And so for most analysis, we still discard 99.9% of the data so far. And when it comes to phylogenetics, that's not the only complication. So for example, there's this nice paper by Morel and colleagues saying phylogenetics of SARS-CoV-2 is difficult. And it's not just because it's many samples. At the beginning, they only had like 8,000 samples, but they already were having problems. The other issue is that branch lengths are short, so many branches will have no mutations on them. Maybe most branches will have no mutations on them. So a lot of the phylogeny is actually not identifiable. And also some of the tools we're used to, like Bootstrap, are not gonna work so well. And the other problem they find is, for example, the fact that the free space, the likelihood values is very rugged, so there are a lot of local maxima. And so finding the best tree is not actually, in likelihood terms, it's not easy. And here we start discussing a few other complications we kind of discovered and addressed in the last couple of years. So I will discuss about the errors in the sequences, how we looked into them, and how we suggest how to account for them. Then we have discussed the evolutionary process of the virus, the mutation rates, and in particular homoplasy, so the fact that some mutations seem to occur many, many times along the phylogeny. Then I will discuss the simulation of genome evolution and briefly recombination, and then in the end I will go back to phylogenetic inference over large genomic datasets in genomic epidemiology. So starting with the errors, so when we look at first the SARS-CoV-2 data, we notice the usual things you notice in this kind of datasets. So some samples are a bit weird. And for example, you can see the sample here as a region that looks like maybe low coverage. And so it's not being called in a consensus sequence, but then just near these low coverage regions, there is a lot of mismatches. So if your typical genome is like 10, 20 mutations away from the reference, if you have a cluster of apparent mutations like these that might not be real, they can be a big issue. So if you look at the ends of the alignment, then this is now the end of the genome, the 3' end. So you can see there are also clusters of the mutations, not just one sample, but in many samples. Apparent mutations, I would say. So it's likely that these are all kind of artifacts of the calling on the consensus sequences, and it's quite easy to take them into account. So you can identify these regions and mask them, or you can identify the samples that have many mutations and also remove them from analysis, or you can entirely mask regions of the alignment, like the beginning and the end of the genome. That's now quite standard. But there are other things that are much harder to spot and to fix. And in particular, if you have errors that are isolated, so they affect only one nucleotide, but they are recurrent. So you have the same error occurring in many, many sequences. Now that looks harder to spot because it looks exactly like a pattern from a normal mutation event with a number of descendants. But if you use a phylogenetic approach, you can infer a phylogeny, you can map mutations on the phylogeny, then you can see that these errors will appear many times on the phylogeny, so they will correspond to many mutation events, and each mutation event will have a small number of descendants. So if you create a plot like this with all possible variants observed in it, and on the y-axis we have the parsimony score, which means for a certain allele, how many time that mutation that brings the allele occurred in the phylogeny. And on the x-axis we have basically the number of genomes that carry that allele. So what we can see is that most variants are down here, so they appear only a few times on the phylogeny, and some of them might have many descendants. But we also see that there are some variants that appear many times, and as a result of many mutation events. And these in blue, they seem likely to be real mutations, just sites where mutations are very, very frequent, exactly the same mutation happening many, many times. But we also see these red ones where we see these mutations are basically associated with a certain lab or a certain pipeline. And so these are extremely likely to be errors in the pipeline. And so with this approach, we can spot them, we can look then at the reads and try to find out where these errors are coming from. And we can recommend masking of these positions. So why we want to do something like that, why we're interested in the problem to begin with, it's because we only have a few mutations in genomic epidemiology. So if you add a few recurrent errors to that, this can really cause problems in the phylogeny, but also in any other type of analysis, genomic analysis that is performed. So for example, in the left here, you have phylogeny with just 2 mutations. So you have 3 clades defined by these 2 mutations. And if you had 2 recurrent errors in these, of course, you're going to have the wrong clades in the phylogeny. So what we need is identify these recurrent errors and recommend people to mask the positions of the genome that carry these recurrent errors. The problem we are facing right now, so this kind of worked until a few months ago, but now we are facing problems because we see that we have recurrent errors at the same position with the same mutations that define the variants at in the spike protein. So in the spike gene. So these are positions that basically are too important to mask and also essential for phylogenetic inference, but for everything else. So we need to find a way to account for these errors without masking portions of the alignment. And this is something that we are working on, and I will come back to that later. So the second thing we looked at is the evolutionary process of the virus, how the virus is evolving in terms of mutations. And first we wanted to look at the neutral mutational patterns. So we focused just on synonymous mutations. And using a phylogenetic approach, we can basically count how many times each synonymous mutation happens. On the phylogeny, we can discard the mutations with just very few descendants to we cannot count them to take into account the effect of errors and selection and so on. And what we see is quite striking. So we see that C2U and G2U mutation rates are much higher than all the others. In fact, about even hundreds of times higher than most others. And so another thing is that basically the evolution of the virus is not stationary. So we have a starting genome, and this genome is getting more and more U-rich or T-rich as time goes by. So So yeah, the mutational process seems highly non-stationary, possibly non-reversible. So this is something you have to take into account when we do phylogenetics, also other analysis. And another effect of this is the distribution of number of mutations is not kind of uniform along the genome, along the type of mutations. So on the right plot, I have basically how many number of times each individual possible synonymous mutation happens. And for most mutation types in different colors here, you have basically no such mutation on the phylogeny. This is the beginning. We had 150,000 genomes, so not like the size of the dataset we have now, but still you have very few possible synonymous mutations actually happening. And if they happen, they only happen a few times, except for C2U and G2U mutations that happen usually happen more than 5 times. And in fact, some of these mutation events happen hundreds of times, thousands of times. So thousands of times exactly the same mutation of the phylogeny. And that's something, again, that we have to take into account, that there are some positions of the genome, and it's about a couple of dozen of them. They're hypermutable and mutate over and over again. And so we need, when we do phylogenetic analysis or any type of analysis, we need to take that into account.
And do you see a clear context sensitivity with showing an ADAR type signature or APOBAC?
Yeah, absolutely, we do. Yeah, that's clearly one of the things. Yes.
Thanks.
Okay, so next issue we face. So at this point, we wanted to run some phylogenetic analysis, run the basically system methods, check what was working, what wasn't working. And the first problem, the other problem we faced at this point was the simulations themselves, because of course, we want to take into account for variation in mutation rate, this non-stationary evolutionary process, and so on and so on, and selection also. So there are software that allow you to have a very general model of evolution. PyBot, for example, is one of them. It's great in this respect, and that's what we wanted to use, but we realized we cannot use it for more than thousands of genomes. So we couldn't really simulate large-scale data sets with it because it's not fast enough. So other software are much faster, like SecGen. It's great, but it doesn't offer you, say, codon models, indels, and it only offers the very basic models of evolution. So we wanted to have something that is that has both benefits. So realistic models of evolution and fast. So we thought, let's make use of the fact that the evolutionary scale is very short, and let's make an ad hoc simulation tool for this kind of genomic epidemiology that is fast, memory efficient, but also is quite broad in terms of model that it allows. And so we developed this software called FastSim that has these properties. We included codon models, indels, hypermutable positions, and so on. So since this is a phyloseminar, I'm going into the detail of the methods. So yeah, let me know if something is not clear. And so the idea here, of course, is to use the Gillespie algorithm. And this is not something new. People already developed simulation tools based on the Gillespie algorithm, which is basically the idea is that you simulate one mutation at a time. So you have a certain rate at which mutations happen, and then when the mutation happens on a branch, you select the position that mutates, and so on. The problem we have is that if you have a large degree of variation in mutation rates along the genome, at each mutation you have to sample the position of the genome that mutates. And you don't want to do this in linear time, in the size of the genome, because the genome is like 30,000 nucleotides. So one way to make this much faster is to use a search tree, like this one depicted here. So this tree is not a phylogenetic tree; is a search tree that has at the tips the positions of the genome. So this is the first position of the genome. This is the second one, and as terminal nodes, and so on. This is an example genome with just 5 genome positions, and the internal nodes represent portions of the genome. So this is the parent of 1 and 2, so it represents the first 2 positions of the genome, and the root represents all 5 positions of the genome. And the rate of the parent is the sum of the rate of the children, and the rate of each node is the total mutation rate of all the descendants. So what we do is we sample a number between 0 and 8.1 here that represent the next mutation, and then we go to the tree searching for the nucleotide corresponding to the number and to the individual mutation event corresponding to that number. So in a logarithmic number of steps in the genome size, we can sample 1 mutation event. Because there is more or less 1 mutation per branch, this is much faster than previous approaches in the context of SARS-CoV-2 of genomic epidemiology. And the other thing is, of course, we don't want to keep track of the whole genome, so we only keep track of the differences of the current genome with respect to the reference. So this, because then typical genome is like 20 more or less substitutions compared to the reference, depending on the time of the pandemic, then that's a reduction in memory demand of about 1,000-fold, more or less. And the really tricky part comes when you want to take into account the fact that the genome is evolving along the phylogeny, and so your search tree for the mutations is also evolving along the phylogeny. So if you have at each step, as you move, for example, here from the parent, you move to a child, you have a mutation somewhere along the search tree, which means now you have a new genome with new mutation rates that you pass to the left child. And instead of creating a new search tree that would take time linear in the genome size, we just add this new path corresponding to the mutation to the search tree. So again, just logarithmic in genome size, and we pass this search tree with this new kind of paths to it to the children nodes of L. So this extended search tree will be used by the children, and then we go back to the left child here, and then we go back to the parent. We forget about we delete the kind of appendages corresponding to the mutations in the left part of the tree. And then we move to the right part of the tree, and you have new mutations, and so on and so on. So with this process, at every branch, every move we make, we only take very small number of steps, logarithmic in the genome size.
Could you use this to simulate context-sensitive mutation rates?
Absolutely, I think so, yes. In fact, we use this for indels, for example.
Yeah.
Of course, everything needs a few adjustments, but I think that should be feasible. And we have codon models, so we have, for example, codon models come with no disadvantage here, in fact,
right,
because of the short scale of evolution, basically.
Cool.
Yeah. So, of course, this was not the only advancement in the last couple of years in this context. I have to mention also ALICIM by the Buimin group, which is very fast, and it also has a broad choice of model. It's not specific for short divergent datasets, so it's not doesn't use this kind of tricks I mentioned for SARS-CoV-2, but it can use the Gillespie algorithm if the branches are short. So it's also very efficient in this context. And I also want to mention, if you're interested in simulating trees, for example, a tree corresponding to the whole pandemic with billions of samples, that's also very challenging. And so the software VGSim can also do that. So I will not go in depth with the recombination. I just want to mention that it's a problem in phylogenetics, in genomic epidemiology. SARS-CoV-2, let's say it's not been a huge problem, but surely an interesting one. And please check out the previous phyloseminar by Yatish Turakia, where he discusses his approach to tackling recombination in SARS-CoV-2. It's a really great paper. And basically, they developed a new approach that is that can scale to millions of genomes, and it's much faster than previous methods. Instead, from now on, I will focus on phylogenetic inference. And the issue is that, as I mentioned before, when we try to use existing established methods, RAXML, BEAST, IQ3, and so on, they struggle when you have too many sequences. Let's say you move to hundreds of thousands of sequences. For most of them is a bit too much. So a very interesting new development has been Usher, again by Yatish Turakia. They discussed this in the previous phyloseminar. And it's a parsimony approach. And it takes advantage of the scenario that we are in, and that is basically short divergence, very, very short branches, to make inference very fast. Another development has been Thorny Beast, which is basically an approximation in Beast that allows you to have something like 10 times larger data sets. But you have to give up some feature of the Bayesian estimation. In particular, you have to fix a good part of the tree topology, basically. And there's also been new approaches on the fractal to parallelize, to distribute the computation of a phylogeny. So what I will be discussing here is the work we have been doing in basically trying to do something like Usher in terms of that phylogenetic approach that would be tailored to genomic epidemiology, but at the same time maintaining the likelihood-based approach that allows us to have large applicability and flexibility, allows us to use evolutionary models, and possibly then also Bayesian phylogenetics and so on. So for now, we are just working in a maximal likelihood context, and we are going to use a lot of approximations within the context of having short branches. And in the next few slides, I will go over all the kind of methodological features in Maple that make it work. So basically, first, how we represent the sequences, then how we represent the likelihood vectors, how we calculate them, how we evaluate different changes in the topology, how we search for such topology changes, and finally, how we update the likelihoods after a change. So all these kind of topics have to be considered in this context. So the simplest thing here is how we represent the genome data, right? So we are used to using something like FASTA, and similar formats in phylogenetics, and we still use them for SARS-CoV-2 for the most part. And this, I think this is really inefficient. This is something that really have to address, and it can be very easily done. So because the genomes are all basically almost identical copies of each other, we really don't need to repeat the same genome over and over. We can use, for example, something on like the one on the right, which is the format we use for Maple. So we have the sample name, and then we have a list of the differences of that genome compared to a reference genome. So if you have, for example, a substitution T, a certain position, you have the nucleotide and the genome position of the substitution. And we can also efficiently track regions of the genome that have not been called. For example, if you have a beginning of the genome, you have a stretch of ends. This can be represented by just the first position of the N and how many ends you have, for example. And same thing for the gaps in the genome. So, and by no means this is a kind of novel approach, but I think it's also not the best approach, but I think it's very human readable. It's much more informative when you look at it, and it's efficient enough to make the problem manageable. So here I'm comparing the size of data sets for number of genomes on the x-axis and the file size on the y-axis. And so I'm showing FASTA in green, and the Maple format is in blue. So you can see there's been these degrees of more than 100 times. Basically, I'm using less than 100 times fewer space to store the data. And if you use VCF file, for example, it's very efficient for, can be very efficient for just a few genomes. But if you increase the number of genomes, then the number of variable sites increases, and then the performance decreases unless you use more sophisticated version of VCFs. Okay, so that was the easy part. But now, of course, when we use maximal likelihood methods, we also need to account for the fact that we have likelihoods. So it's basically a similar amount of data we need for the genomes, we also need for the likelihoods. And usually what we have is for each node on the phylogeny and for each position of the genome, here, as an example, I have only positions 1 to 10. We have the likelihoods of the 4 nucleotides here in the 4 columns. And so somehow we need to compress this information. And what we do is basically, instead of considering likelihoods as we normally do in phylogenetics, I'm considering relative likelihoods. So I basically normalize every row of this matrix so that the sum is 1. And the other trick is that, so there's not much advantage to that, apparently. But the other thing is that for most of the genome and for most of the nodes in the phylogeny, the likelihood will be highly, highly focused on just one nucleotide. And for the vast majority of the time, then that nucleotide will be the reference allele at that position. So, for example, here for position 1 to 9, I always have the reference allele where the likelihood is highly focused. And so what I do is I discard the information in the other cells of the matrix at these positions, and I just store the information that from position 1 to 9, the reference allele is the most likely one by a very large margin. And if there is uncertainty here, so there are 2 nucleotides at position 10 that are similarly likely, let's say the difference in likelihood is above a certain threshold, then I do the likelihood calculation as usually is done in maximal likelihood phylogenetics. So that's treated normally. But now, because the number of differences with respect to the reference is going to be always just 1 or a few dozens of positions, then it means for the vast majority of genome position, I will be in this scenario here, that I can represent them with just with an R. And this greatly reduces the memory demand for storing the likelihoods, but also reduces the time for calculating the likelihoods. As an example here, I have 2 child nodes where the likelihood for position 1,9 is concentrated in the reference allele. What this means is that the likelihood for the parent node will also be highly concentrated in the reference allele for the same positions. Because the only alternative here is that you have basically 2 mutation events on these 2 branches, away from the reference allele, away from another allele. And because we are assuming the branches are very short, this is extremely, extremely unlikely. So we can ignore this possibility and just keep track of the fact that the likelihood is concentrated in the reference allele. If in any other case where there's some discordance or there's some uncertainty in the children nodes, then we perform normal likelihood calculations. And if the parent node likelihood will not be focused in one nucleotide, then we store the 4 likelihood values. And this is all nice and simple, but actually there are a number of complications to consider. And these do not impact basically the computational demand at runtime, but when implementing the method, that's something that you need to consider. To give you an idea of the complications, so here you have 2 genomes for 2 samples, 1 and 2. And the first one is 5 N's, then you have a number of A's, and I'm assuming the A is the reference allele all the time. And finally, you have a T. And you have some more or less the same, just that the stretch of N is short for the second sample, and you have no SNP at the end. So what do is we want to calculate now the likelihood vector for the parent node, taking into account the branch lengths, L1 and L2. And if there is no information in the 2 children nodes, so the first 3 nucleotides, both children have N, so we also store an N state for the parent node from position 1 onward. From position 4 onward, the situation changes because you have a first sample is N, but the second sample is a reference allele. So what we do is store the reference allele in the parent node, except that this time we have to take into account that the observation of the reference allele is not at the parent node, but it's a child node. And this is because so there's no information on the left branch, but on the right child, you might have a mutation. So if further on the phylogeny, you observe another allele, there might have been a mutation event from this other allele to the reference on this branch. And so we have to take into account the length of these branches from the node to the observation of the allele, and basically pass it on to the further parents nodes. So this goes into an additional kind of parameter in the likelihood vector. If both children nodes are in type R, are reference alleles, then the parent node will also be in the reference allele, and we don't need to keep track of these branch lengths anymore, because we can consider the reference allele as observed at the parent node, because the probability of having mutations in the child branches is very unlikely, as I mentioned before. In any other scenario, basically, we are doing normal likelihood calculations, and we are storing these. So this is now the last position of the genome, and we are storing the 4 likelihoods in a typical likelihood vector. Okay, so we have a very fast way to store the likelihoods, to calculate the likelihoods. Now we need to evaluate the tree changes, and as I mentioned before, the problem is we're not having the full likelihoods, the normal likelihoods, we are having relative likelihoods. However, it turns out you don't need the total likelihoods to evaluate the tree. So usually what people do is they evaluate the value of a tree, calculating its total likelihood. However, we can bypass that step, because we can simply calculate the contribution to the total likelihood score that making a change to the tree gives. As an example here, I have, say I have a tree and I remove a subtree from it. So this one on the right is a subtree that I might want to reattach on the tree somewhere else. And so I have relative likelihoods here and the root of the subtree. And in the rest of the tree, I have total likelihoods, so the likelihoods for all the data in this remaining part of the tree that I pre-calculate. So what I can do is I can compare the likelihood here at the root with the likelihood in these other nodes of the remaining of the tree. And I can calculate, by comparing these vectors, I can calculate the cost of appending the tree at that new position in the tree. And this cost can be calculated, again, very efficiently. I can basically count the mutations. Each mutation is a certain likelihood cost, which is approximated like this in the logarithm of the branch length times the mutation rate. So that's basically a first order approximation of the likelihood cost. Again, due to the fact that we are assuming the branch lengths are short, then we can do this approximation. And similarly, if there is a stretch of the genome with reference allele in both ends of the branch, then we can very quickly calculate the cost of having no mutation on this branch in this region of the genome. This is done again, basically approximating the logarithm with the first order, and then multiply branch length by the total submutation rate at this stretch in the genome. So this can be pre-calculated, more or less, so that basically the cost of calculating the probability of no mutation here is just constant, no matter how long is the stretch of the gene.
I think I know the answer to this, but maybe you can just explain and give some intuition why it's fine to use the partial likelihoods for the subtree, but you need the total likelihood for the rest of the tree.
Yeah, so for the subtree, we basically, okay, the two things are almost the same in the sense that here we don't want to account for the root probabilities. And the reason is that then we are reattaching this somewhere else in the tree, but not as the root of the tree, right? While here for the remaining part of the tree where the subtree is going to be attached, we need to take into account all the data in the subtree, like we are doing for this one, but we also need to account for the root frequencies, because the root of this tree is going to be the same, basically. So that in the end, basically, so basically, because these are normalized, another thing maybe I should mention, because this is normalized, the normalization factor here that I'm not including is basically the laclude of the subtree, the product of all the normalization factors. And likewise for this rest of the tree. So in the end, if I had these normalization factors, I could calculate the total laclude of the tree this way. But that's not necessary, because no matter what step I take, these normalization factors are going to be the same. So this doesn't help me to identify the best new location for the subtree. Probably making a mess. Sorry.
No, that makes sense. But that's so. Let's see though. Once you do find a good place for this subtree, you have to recalculate total likelihood vectors. But I guess
I'm going to go there. Yes.
Oh, okay. Sorry.
Actually, the other tricky part, of course, I completely skipped here. The other tricky part is when I remove the subtree from the tree, I need to update the lacludes here in the rest of the tree, because now that subtree is not there anymore. So the lacludes change.
Yes.
But that can be done very efficiently, and I'm not exactly doing that because I update the lacludes as I move and I traverse the tree. So that can be done very efficiently at no basically additional cost.
Okay, cool.
So now this is the part of the algorithm basically where I traverse the tree to look for the best basically SPR move or the best placement for a new sample. And this is the part of the algorithm where actually I have something more demanding than what people usually do. So usually people start at one place and look in a certain rate. If the current placement of this subtree or sample is the root here, they look in a certain neighborhood of the phylogeny for a possible new placement. But for SARS-CoV-2, it doesn't work so well because of the complexities I mentioned before. It usually needs a much deeper exploration of the phylogeny to find the best location. And probably in large part, this is due to the homoplasies I mentioned before, the same mutations occurring over and over. But part of it is probably also due to the fact that some samples have missing information. Part of the genome is basically not called, and so on and so on. So what I'm doing is I'm exploring not all of the phylogeny, just part of it, but I'm following the laclude score as I move. So let's say the initial placement has cost of 70 log laclude units. I move to the left child first and left child to the left child and so on. If I see that the laclude score of these new placements gets much worse, then I stop and I don't explore the children of that node. Likewise here, I stop exploring. But if I see on the right side of the tree, now I have a much more promising laclude scores, I keep going in that direction. And as long as the laclude scores improve or they don't decrease too much compared to the best I find so far, I keep exploring that part of the tree, basically. And so this is substantially more demanding, in particular if you have certain samples that are very hard to place and so on. But in accuracy terms, it is worth it. And so what happens after I find a nice spot for my sample or my subtree? Now I have to attach it to the tree, and as Eric was asking before, we need to update the lacludes, right? And here's the other trick, the other convenience to have relative lacludes, that total lacludes can be affected by events far away, but relative lacludes are not. So you can think of them as basically posterior probabilities of the nucleotides. And so if somewhere far away in the tree you have a new mutation added, that's not going to affect the posterior probabilities of the nucleotides in some other region of the tree far away. So we can take advantage of that, and we can start updating the relative lacludes at the position that are affected by the change. And then these changes are kind of passed on the phylogeny as you move. And at some point, you reach a point, you can compare the old lacludes, the new ones, and you see no change. So you basically stop the updates in that direction. And so in the end, you only traverse a very, very small area of the tree. So updating the lacludes is very, very efficient.
Sorry, I thought though that you would need to have, don't you need your the total likelihoods?
Yes, yes, but those are also relative. So they're also, so when I say total likelihoods, I mean the likelihoods take into account all of the data, but they are still relative likelihoods.
I see. Okay.
Yeah, sorry, very confusing. Okay, so all of this put together, what does it mean? It's we implemented this approach in Python because it was convenient, and we are using PyPy3 to make it run fast. And so what we can see is that the memory demand here on the right is reduced by a large margin. So what we get in the end is more or less similar to Azure. And on the left, the time is also quite improved. Sorry, let me mention different methods. You have Maple in blue, and you have Azure, and basically MatOptimize is a addition to Azure that improves the phylogenies. So these are parsimony trees inferred by Azure and improved by MatOptimize. So you get kind of similar runtime between Maple and Azure in this context, and more or less similar memory. And the other colors are RAXML in purple, FAST3 in green, and IQ3 in red. So if you compare, for example, to RAXML, you have a huge improvement in time demand, also some improvement compared to the other methods. And in memory, much more, because the difference seems even to increase as the number of samples considered increases. And the interesting part is that because we are doing this deeper tree search, despite the fact that we are using approximations here and there, we actually get better trees in the end. So on the left, we have results from simulations. On the right, we have results from real data. Colors are the same as before. And on the y-axis here, we have the Robinson false distance between the estimated tree and the true tree in the simulations. So basically, the y-axis tells you more or less how many errors you have in the tree. So lower is better. And in the right, you have the real data. So we don't know the true tree, but we can estimate the likelihood. We can estimate the likelihood of the inferred trees. And here I'm inferring the likelihoods in IQ3 in the end, so that the likelihoods are always comparable between methods. And what the values are here are the difference in log likelihood score between the best phylogeny for that run and the others. So they are all 0 or negative values, and more negative values mean less likely phylogenies estimated. And so you can see that the difference in likelihoods can be absolutely massive, in particular compared to FAST3 and IQ3, and quite substantial also to RAXML, even though here we could only run RAXML up to 5,000 sequences due to our computational resources. And so Maple is always the one giving the highest likelihood and the smallest number of errors. But what's also striking is that parsimony is not necessarily worse than likelihood methods. So basically, on real data, Azure MatOptimize gives you higher likelihood trees than maximum likelihood methods, apart from Maple. And the most likely reason I can think of here is that basically Azure does something similar to Maple. It does much deeper tree search. So even though he's using parsimony, in the end, it explores the tree space more, and it gets better trees in the end. Okay, so yeah, sorry, I forgot to mention, I also have other simulations. In simulations, you have different results, but if you compare Azure and the other methods, and that's because simulations are quite different from real data. So basically, simulation datasets are much simpler to infer, and more cursory tree search is enough. So we run also other simulations where we introduce hypermutable sites, where we introduce basically where we mask some of the genomes as to mimic the real data, and so on and so on. So depending on the simulation scenarios, you can see different performance between the methods, but Maple is basically constantly the most accurate approach. So other things we added to Maple are, for example, online inference, where we allow the user to load a tree, pre-inferred tree, and add new samples to it. And it's more efficient than just rerunning the phylogenetic inference from scratch. And that's useful for a scenario like SARS-CoV-2 because we have new samples coming in, say, every week, so we don't want to run the inference from scratch. We have a number of parameters that we can use to make the tree search deeper or more shallow, depending on how quickly one needs an answer. And we have, so far we only have nucleotide models, but we have the broadest range in the sense that we allow non-stationarity and reversible substitutions. And we now can also infer rate variation. So in particular, we allow each position of the genome to have its own rate, and we infer the rate for each position. And we do that using expectation maximization. So it's quite numerically stable, and it seems to work very well. And finally, the things I used for the benchmarking, for example, to evaluate the allelacludes for very large trees, or to calculate the Robinson faults for distances for very large trees. These are also included in Maple now, so you can use the same tools as I use if you want to compare large trees. So we have a preprint that you can check out with more details, and we are working on a few other things. Currently, we are working on modeling sequence errors. This is to address the problem I was mentioning before. We have these positions. We have recurrent errors that we want to take into account. So one way to do that is to model the error process at the level of the sequences and to model position-specific error rates. So we are hoping to use this approach to improve phylogenetic estimation in this context. And also, our collaborators are working on a C++ implementation of Maple that would have the advantage of being very highly parallelizable, because so far we don't really allow that in our Python version. And regarding future work, there is really so much I would like to work on and extend. So let me know if you are interested in any of these. So basically, I would like to extend the approach to time trees. And in turn, this will allow us to work in the Bayesian context like BEAST and to allow phylogeography and phylodynamics, for example. We would like to basically study what kind of approaches we can use to measure uncertainty in the trees, since the bootstrap doesn't seem to work very well. So we need to assess what kind of approaches we can use instead. And we think we could extend Maple to model selection, in particular at the codon level, that say typical codon models that should be relatively easy and computationally efficient, possibly recombination. And another thing that would be nice would be to model indels. By using the fact that the evolutionary scales are very short, it should be possible to infer the alignment and the phylogeny at the same time, basically by integrating over possible alignments while you're doing the placement of the samples. So I think this is highly feasible, but of course, it's something that would take time to implement. So let me acknowledge my collaborators that made all of these possible. And I would give a special mention to William, who did a large part of the FastSim implementation, to Mirte, who worked on the error models, and to Nian, who is working on the C++ implementation, and all the others have really been very, very helpful. So thank you very much for listening, and please let me know if you have any questions.
Awesome. Thank you very much, Nicola. That was very interesting. I invite people to ask questions on the YouTube live comment box. So I'd love to know to what extent you could turn Maple into a library, like the Maple likelihood evaluation into a library that's morally equivalent to Beagle. So I think, I assume you know, but Beagle is basically has an API that allows you to do fast standard likelihood computations with an API that sort of says like, here are the types of operations one typically does when you're doing likelihood-based phylogenetics. Like you're at a node and you join two nodes together. You need to like do some marginalization and so on and so forth. So, and evolving things along branches. So to what extent could you do something like that here? And it seems a little bit, so for instance, like Beagle does not have a notion of a tree. And that's an explicit design choice that they made, and that's important because Beagle gets integrated into Mr. Bayes and Beast, and they all have their own notions of trees. And it seems a little bit like the maple likelihood is more intimately tied with the tree structure.
Yeah, so there's one factor here that most of the things I mentioned are really useful when you have short divergence, right? So that's for any application of most of the things I mentioned so far, that's really the only application. And for long branches, most things will break down or will be extremely inefficient.
Right.
So that's one limitation, but it's probably not the main limitation. Let's say that because part of this could be made into a library, but it wouldn't be so easy then to kind of integrate this library with existing tools, for example, because it's not just one change, right? It's not just the representation of the likelihood, it's many things, how they're calculated, but also how they are used. Yes. So I think part of this thing, could be made into a library probably. But I don't think it would be easy to necessarily make it work with basically what's already there, right? To integrate with what's already there. And maybe, there's another aspect I'm thinking is that there are some parts or the things I've been working on that actually could work more in general. And this is basically the relative likelihood part. So that part could be implemented like that, as you mentioned in the library, but that would also require then to change not just the likelihood calculation representation, but also the part of the rest of the code. So this could be a project, but I also feel like maybe it's a bit too much in infancy to be useful in a sense, since I'm the only one who's using this kind of thing so far. But I'm really
but at the same time, if you were gonna, I mean, you're talking about integrating into Beast, right? I mean, so you already be converting all of your code into Java.
But the thing is that I think it wouldn't be so straightforward to just reuse the same thing. I'll just reuse the code as it is to make it work in Beast. I think it would anyway require rethinking how the MCMC moves will work. So because of the relative likelihoods, basically. So it might be possible, but it's not something that I have a clear path about,
Right. On your benchmarking results, I feel like I noticed that, I mean, in a sense, like you're not running Mat Optimize, and Mat Optimize is sort of in its area of strength, right? Like, if I understand correctly, this is something, either the simulations and the real data, it's sort of like a subset of all of the data. Whereas, like, I think the idea of Mat Optimize and Usher is that you run it on a million sequences or however many million sequences. And so that it seems like that's a slightly different parameter regime than it was designed for.
So you're saying that, okay, it's not the ideal application for Usher and Mat Optimize. In some sense, it's true for Usher because Usher was meant really for adding samples to a given tree, to a large tree. It's a placement tool. But like now with Mat Optimize, it basically means that you can get the initial tree with Usher this way and optimize with Mat Optimize, and it's going to be really good, as you can see here. So I really wanted to add this to the comparison for fairness, because I knew it was going to be good probably. And then if I had, so the largest data set I'm comparing to is like 200,000 sequences out of a data set to 500,000. And I think so. I'm basically trying to apply also the method to the whole data set. Basically, that's the idea. And I get similar results. So I think that I'm not really sure what kind of drawback you're thinking about. Maybe because of the branch length will be longer. If
Right.
the
Exactly. Yeah. I mean, so
it's not such a big deal. Like I think once you have, say, a few thousand sequences, when you add more and more samples, the branch length added would be, 0 most of the time.
So you're saying that even when you have that large data set, if you add additional sequences, you won't break up existing longer branches?
So this is the thing I was mentioning, basically. So these are the Robinson files on larger data sets. So hundreds of thousands of sequences. I think I basically already had this before, actually. But basically, even if, 200,000 sequences now, this is, Asher does get better. But the pattern is the same, basically. So I don't think this is having a huge impact.
Okay. In your error model, do you one thing that that I've heard happens a lot is reversion to reference when you have people who are over aggressively calling reference when they have ends. Does it include that?
We have not. So far, we're using very simple error models. That's what we want to do. We had not really thought about it, how to do it, actually. So far, we were thinking of having just position-specific error rates, maybe nucleotide-specific error rates. So that might take into account having position and nucleotide-specific error rates. But explicitly modeling the reversion to the reference or to another allele, if the other genome is more or less common in the population, that will make things more accurate, but they will be also more complicated. So again, I haven't really thought about how one could go around it.
Okay, cool. Yeah, I mean, yes. I've heard this is like one of the major issues that people run into.
Absolutely, that's the main bummer these days.
Very cool. I know you need to get your bus to head home, but thank you very much for an awesome talk and for wrapping up our session. And I should also acknowledge Nicola for helping me think of all the folks who populated this great session. So thanks on both counts.
Thank you very much again, Eric.
Bye.
Bye-bye.